Hello, this is a video lesson on molecular and cellular radiobiology and dose-response relationships. Let's begin by talking about the direct and indirect effects. The direct effect is where the radiation just strikes DNA and causes damage. It happens with a direct action. The indirect effect is also called the radiolysis of water, and it's where radiation strikes water, a water molecule specifically, and forms ions and free radicals. The free radicals seek out DNA and then strike the DNA. So it's an indirect action. So the indirect effect, which is called radiolysis of water, is the interaction of radiation with water and which forms ions and free radicals. So in the case of this process, we're going to form ions which have either positive or negative charged particles, HOH plus or HOH minus. It could be H plus, OH minus, but these are ions, where free radicals are going to have signified by an asterisk. The free radicals are highly reactive, they're unstable, and they exist with a lifetime of less than 1 millisecond. They're capable of diffusion through the cell and interaction at a distant site. They contain enough energy that can be transferred to other molecules to disrupt bonds and produce point lesions at some distance from the initial ionizing event. There are three reactions that can occur. So the number one, you can have ions which are just positively and negatively charged atoms. They can recombine to form a stable water molecule with no damage occurring. A second perhaps item that can happen is a free radical can migrate to a molecule such as DNA and cause a hit. And if this happens, it causes biologic damage. And the third one is free radicals can combine to form a toxic substance which migrate to the DNA molecule and cause a hit, and this also causes biological damage. The principal effect of radiation on humans is indirect, and the reason why is because 80% of the body is comprised of water. Less than 1% is DNA. So there's going to be a whole lot more interactions which are indirect than direct. And the indirect effects are amplified because of the formation of free radicals also. Irradiation can happen in vitro, which is outside the body, and irradiation can happen in vivo, which is within the body. And molecules are more sensitive when irradiated in vivo because there's water, there's oxygen. We've got things that can increase sensitivity. Irradiation of macromolecules involves one of three things, which is main chain scission, and that's the breaking of a backbone of the long chain macromolecule of DNA. Cross-linking, which is side structures with a sticky substance, so they attach to a neighboring macromolecule. Or third, we can have disruption of a single chemical bond, which are called point lesions. For radiation responses of DNA, we have 5 of them. The first one is main chain scission with only one side rail severed. B is main chain scission with both side rails severed. C is main chain scission with subsequent cross-linking. D is main chain scission causing separation of bases, and DE is a change in or loss of a base. Let's take a look at these. In A, we have one side rail that's been severed, so that's called main chain scission of one side rail. In B, we have main chain scission of two side rails, which is actually can break the DNA in half. In C, we have main chain scission with cross-linking, where another DNA macromolecule is joining the other one. And D doesn't have the best picture, but it's separation of the bases, breaking of a bond, separation of the bases. And the last one is change or loss of a base, which is showing in E. And this is changing the triplet code and may not be reversible. This may also be called point mutations. Now, DNA is the most radiosensitive molecule in the body, and it's the most important. The reason why is it controls cell function. It contains hereditary information if it is a germ cell, and it comprises less than 1% of the molecular makeup of the body. So since it's so critical and such a small component of it, it is very important to us. DNA makes up chromosomes, which makes up cells, which makes up humans. A chromosome contains miles of DNA, so if there's a visible aberration that appears, it signifies a considerable amount of radiation damage to the DNA itself. So severe radiation damage to DNA that can be visible under a microscope can be detected. A is a normal chromosome after the S phase, and B is referred to as terminal deletion, otherwise called chromatid deletion. C is called dicentric formation or dicentric chromosome, and D is called a ring chromosome, otherwise called a ring formation. But that's where we have severe damage to DNA. Three observable effects from irradiation of DNA include malignant disease, cell death, and genetic effects. The cause of stochastic effects are the damage to the DNA that results in abnormal metabolic activity. So its uncontrolled rapid proliferation of cells is the principal characteristic of radiation-induced malignancy. Stochastic effects have a linear non-threshold dose-response relationship that we'll talk about in the next section. The cause of deterministic effects are that we have damage to DNA that can result in cell death. If enough of the cells of the same type respond similarly, then the tissue or organ can be destroyed. And this is a nonlinear threshold dose-response relationship, which results in cell death. The third one is the cause of genetic effects, which is going to be damage to DNA which occurs in a genetic cell, and that's response to radiation exposure will not be observed until the following generation or even later. And this has a linear non-threshold dose-response relationship. The target theory is the concept of a key sensitive molecule, and that molecule is DNA. In order for a cell to die following radiation exposure, its DNA molecule must be inactivated. DNA does not look for DNA in cells to destroy them, it interacts in a random fashion. Now let's look at part 16E on dose-response relationships. We have two really important dose-response relationships that are shown in this diagram here, where on the left-hand side we have the linear non-threshold we'll talk about, and on the right we have the non-linear threshold. So relationships can be linear or nonlinear. On the left we can see linear relationships with A, B, C, and D. On the right, we can see nonlinear relationships, which are not straight lines, and that's what nonlinear means. Threshold versus non-threshold, A and B on both sides in blue are both non-threshold. They both start at 0. They both have that there's no dose that the dose is 0, and before you see a response, and then also the green or the yellowish colors, those are both threshold. They have DT, and that stands for threshold dose. And that's where you have to have a certain dose before you see a response. An example of that would be erythema, where it takes 2 gray of radiation to the skin to cause erythema. So the definitions are linear is that the response is directly proportional to the dose. When the radiation dose is doubled, the radiation dose to radiation is doubled as well. Nonlinear would not have that straight line appearance. Non-threshold is any dose is expected to produce a response and it begins at 0. The dose is at 0. Not threshold means that we've got a dose at which a response to an increasing X-ray intensity first occurs. What that means is the dose is above 0 for where the dose begins. Below a threshold dose, no response is measured. The example I just gave was erythema. Another example is epilation doesn't begin until at least 3 gray of radiation. So a person has to have 3 gray of radiation collimated down to a small area before epilation occurs. A natural response level indicates that even without radiation exposure, the response occurs. So taking a look at the picture here, the RN is a natural response level. So A is a linear non-threshold dose-response relationship in which there is a natural response where there was a response already without a dose. B is a linear non-threshold because it's a straight line and it starts at 0, so it's linear non-threshold. And both C and D are both linear threshold. They are both linear responses, but they have threshold response, threshold doses. So there's a certain dose before you see a response. A in this picture and B are all both non-linear because they're both neither one lines, so non-linear, and they're non-threshold because they start at 0 for the dose. In the green curve, which is C, that would be a nonlinear threshold. So nonlinear threshold means that is not a line, but there is a threshold dose, which is a dose above 0. So if we take a look at stochastic responses, stochastic means that we're going to follow the linear non-threshold dose-response relationship. It follows a low radiation exposure, appears as a late radiation response, results from cells that survive an initial radiation and retain a memory of the event, and there's no threshold. It starts at 0. Any dose can cause a response, so again, it starts at 0. The chance or probability for response increases as a result of increased dose. And examples are cancers, except for skin cancer, and genetic effects. So again, stochastic effects follow a linear non-threshold dose-response relationship. Deterministic effects follow a high radiation exposure and appears in early response. There is a threshold, so it's non-linear, but there is a threshold. And it's an S-type or sigmoid type is the other name for this curve. As dose increases, the severity of the response increases also. And examples are erythema and epilation. And that brings us to looking at stochastic and deterministic effects. Stochastic effects are those that follow a low dose and they have a late effect. We have any dose can cause a response starting off at 0 for the dose, and that the response is going to be linear. Leukemia, cancer except for skin cancer, and also genetic effects are going to be those that are types of stochastic effects. Secondly, we have deterministic effects in which there is a non-linear response and there is a threshold, so that there has to be a certain dose before you see response. An example is permanent sterility. It takes 5 gray to cause permanent sterility, and unless you have 5 gray, you're not going to see permanent sterility. And this concludes our video lesson on this topic.